Yes, today I will talk related with microbial consortia. I will show one tool that was developed several years ago in our lab, FLYCOP, Flexible Synthetic Consortium Optimization. So first of all, I will explain why we needed that. As you can imagine, when we study the nature, we can start with only one species, only one cell. That is the easiest way. But if you started to see that this cell is growing with others, creating populations, start to be joined with even other strains, living together like a sorbar, or even with other species creating microbial consortia. So the biological complexity correlates perfectly with the phenotypic variability. It's something that we have it in mind when we are studying the nature. So this is the reason because in biotechnology, first they started with monocultures to produce metabolites. They use it a single strain that with modification, engineering, adding new genes from a specific substrate, we can produce a desired product. But we have it a lot of problems when we do it in that way. Why is that? Well, in the moment that you have it only one species with a lot of modifications, you increase the metabolic burden with the number of genes that you have it. It's less robust than in nature because it's more sensitive for changes, for stress, and it's not always able to produce a complex task because in the process to generate these products maybe can appear toxic metabolites that can reduce the productivity. And this is the reason because the biotechnology started to use it synthetic microbial consortia. Try to join multiple species, dividing, for example, the work in different tasks to solve it this. Because in that case, when you have it multiple species, you can do it the division of labor. That is super important for that. You can divide it a pathway, for example, in multiple species, dividing the work, reducing the metabolic burden, increase the robustness because they work separately as well, and even you can expand the metabolic capability of the consortium. So, in biotechnology as well, is well known to use the DBTL cycle, that is design, build, test, learn, that in general is to perform several cycles to improve the or engineer the species that we wanted to use for biotechnology. If you have it more than one species, you have to increase the level of complexity in this DBTL. You start, of course, to optimize genes for the specific function, later the pathway, later the chassis that has to be included in the pathway, and at the last one, the interactions between the species that you wanted for the microbial consortium. So the number of the variables that you should to test it is much, much higher than a single one. We can, of course, try to engineer this this microbial consortia in a most experimental way, like for example, evolutionary experiments. That doesn't matter what's happening between the species, you create an stress to obtain a goal. Of course, if you use omics, you can infer the interactions between the species based on concurrence. Or even if you have more information, you can deal with genome-scale metabolic models that you can identify the metabolites can be shared between the species. So once that you have the interactions, it's necessary to engineer them. Again, from the experiments, you can obtain previous knowledge from the literature. You can identify metabolites from them, and maybe if you remove one, start to sharing other. You can again use it omics that you can infer the genes that are activated, and you can infer more or less the metabolites that can be shared between the species. Or again, we can use it the interactions from metabolic models and try to mimic the reality. How to perform the derivation of labor? That is one of the main engineering process for the species. We can, of course, try to use metabolites that are co-feeding between the species to be controlled one each other, or maybe we have it heterologous pathway we can divide it between this network in different species to finally produce the final metabolite. Or even we can distribute the catalysis in different species to reduce again the metabolic burden. So in this point is where we have it a lot of problems to try to design so many of variables. Because of course we have it other goals. We wanted, of course, production. Maybe we can want to have a problem with the pathway distribution. We have it the community stability along the time. That is important to avoid that some species dead very early comparing with the others, or even to try to identify the best medium for all of the species. So the idea is we have the idea in the lab to use metabolic models to try to infer these variables to identify the best ones. But the problem of metabolic models that I will explain what they are is are limited, and we had to extend this functionality. A metabolic model, of course, is a mathematical way to try to mimic the phenotypic results from a species based on the reactions that are related with the genes of an organism. We have the reactions. We know how the metabolites are related with the reactions, and this is the reason because it creates a matrix, a stoichiometric matrix. And thanks for flexible analysis, we can identify the optimal solution that in general is the growth of the species for a specific medium at the beginning. So we are going to use this to try to simulate and mimic the behavior and phenotype of the species. But this is only for one. We need to do it for several of them. And here appear FLYCOP. So once that we have the multiple genome scale metabolic models together, we need to configure them. How to do it? Well, there are tools for dynamic FBA, that is, there are simulations based on FBA that optimize the growth along the time. You can obtain, of course, in several points of time. We can obtain the concentration of the species. And we use these tools like Comets, that is the one that we use in our pipeline, to try to infer the best ratio at the beginning. That is something that is we can control it in the lab, the ratio between the species in the inoculum. We can control as well sometimes the uptake of any specific elements in the medium or the production rates. So this is something that we needed. And how it works FLYCOP to infer them? We have multiple configurations. We have it a large sampling elements that we have it, like, for example, the ratios, initial biomass, and we use comets to simulate the metabolic models using these parameters several times. And SMAC, that is the third element of the pipeline, is an optimization process that try to identify the best ones to increase the goal that in general is the production of your metabolite. Continuous is continuously a loop, is limited based on the number of times that you wanted to search, and at the end you will obtain the best configuration based on the fitness. So this is the original version that it was updated several times, and now is under developing with the second version translated to Python. That previously was mainly Shell scripts and R. And now we created several classes to create a framework where we have it the option to have several optimizers, not only SMAC3, for now SMAC3, but can use OPT-UNA. From simulators for now we have it Comets, but as well we can use it the classical dynamic FBA algorithm that is a little bit faster than Comets. And this is the first point that we started. So the idea to use FLYCOP is first, of course, you needed the gene modifications that you wanted to mimic the reality of your problem. So, and when you are using genome-scale metabolic models, if you have it, for example, mutants, you should to remove it, the reactions associated with the mutants. Or if you know it, for example, the uptake or the production rate of a specific metabolite, you should to add this information for the metabolic model uh, previously to use FLYCOP. The second one is the definition of a scenario. This is one step that is important and it's complex because here is necessary for the user to know it programming skills based on comets, to programming a scenario that you wanted to mimic the dynamic, and at the end, FlyCop will run the optimization and will iterate several times until find it the best solution. That is important to mention that not always will be the best one because, of course, we are try to looking for in the global solution space. That is for time is totally impossible to identify. So for now, what we improve it is FlyCop based on Python and the new versions of Comet 2 and SMAC 3 decrease a lot the time. So to check it that we selected randomly several genes and they work it eating glucose and minimal medium and try to identify the best solution for total biomass of them. We could, in this case, to use it in much more models than previously. That is important because, of course, previously you can use it as small microbial consortia, but maybe you need it much more. And of course, it's require less time than before. But in that case, it was because the problems that we have it for non-parallelization that it was doing previously. So I will show you a small use case that we did it. In that case, we use it a small microbial consortia where we have it a Cynococcus, that is a cyanobacteria, that can use the light and the CO2 to produce biomass. But in that case, it's interesting to use this cyanobacteria to produce sucrose that can be used as carbon source for the second species. We will use Pseudomonas putida that we use in our lab to accumulate PSA. Of course, we need it now to optimize this microbial consortia. And in that case, we'll use FlyCop. So what we had it to do in advance? We have it as Cynococcus that for simplification the model, I can mention that the model can use it different intensity in the light to model the growth. In that case, we simplify with only one to reduce the time of running. And we overexpress two genes involved in the sucrose secretion under stress, so to try to mimic the reality. The second model that is Pseudomonas putida is the species to eat the sucrose. We needed to include a gene, an invertase, to include the sucrose to divide it later in glucose and fructose. We had to remove the nitrate assimilation system because the accumulation of PSA in bacteria require a nitrogen starvation. And the medium that requires Cynococcus to live require nitrogen. So in that case, the medium will contain nitrate that will not be used by Putida to avoid this problem. And later on, we simplify the PSA monomer production. The model contains a lot of type of monomers for PSA. So in that case, to simplify, we wanted to know it only the production of PSA and doesn't matter which one. Which parameters we can optimize in that system? Of course, the biomass of Cynococcus, the initial biomass of Putida, but as well the sucrose secretion rate that we don't know in advance and depends of the overexpression of the genes that we have it, we can increase or decrease. And the concentration that at the end we need it to simulate the nitrogen starvation. And of course, the fitness is the accumulation of PSA, in that case, 100 of hours, because the system is continuous. As you can imagine, in the moment that you have it live, the Cynococcus should produce sucrose all the time. So if we don't stop it, it will continue. So the medium will not be almost finish it. So after those modifications, we simulate with dynamic FBA and FlyCO try to identify the best solution. The solutions that we identify are quite similar between the new version and the previous one, so that's something that it was nice. It's a little bit better in terms that is fine-tuned versus the previous one, because the previous one only works with discrete numbers that can identify with with a simulator, and in our case we can use all the time real numbers and with a large number of decimals. So this is the reason, because there are a small difference, but at the end it is quite similar. The way to code this new version is quite simple at the end. The most complicated one, of course, in that case is with this short code you can run it. It's just create the object for FlyCop. You can create the simulator that you wanted. In that case, we use comets. You loaded the consortia and include the simulation params that you wanted. In our case, for example, it was the initial biomass of Synechococcus, the initial biomass of Putida, and so on. And you create the scenario. This is the longest the PSA scenario, depending of your simulator. If it's Comet, you should to fill it one function with the simulation based on Comet's framework programming code. And what is the output? At the output, you will have, of course, the best solution that you will have it, and as well, it's nice to have it the rest of the points that you were searching to understand if you can identify correlations between different variables that you are using. For example, here. We can see that, for example, of course, the PAC production is is correlated with the carbons that you have it. That is quite expected, but you can see as well that the most of the points that it was identified by FlyCop always tell that you needed much more Synechococcus biomass at the beginning. If you see the second column and the density line that you have it. And of course, for Putida, doesn't matter. It's better if you have it more, but it's less important than Synechococcus. It means that you really needed to design your microbial consortia a large amount of Synechococcus at the beginning to produce the sucrose, and at the end for Putida. What else we can see? And of course, you needed the most of cases a low concentration of NH4 that it was expected at the beginning, because it's supposed that with low NH4 the correlation with PSA will be high, because the PSA is only accumulated when you have it a low amount of nitrogen. We try to use it for a second use case. In that case, it's for a biolacey in production, that is anti-cancer, antimicrobial compound. That several years ago it was used in a microbial consortia with two E. coli, where they have it a tryptophan production for one, and this tryptophan is transformed by an operon, the violin operon, to violacine. It's problematic to put everything in the same E. coli, because it's quite stressful, this process, and the authors divided into. So we use this example to try to see the result that they have it comparing with the FlyCop was doing. They as well trying to optimize the medium, selecting multiple carbon sources. So in that case, we need to modify the species based on the strains that they use it in the paper. In the paper, they have it one E. coli that has a higher tryptophan synthetase activity compared with a wild type. So we modulate to increase the flux about the tryptophan synthase. We include the heterologous pathway inside the biolosin production strain that we should to include, of course, the reactions inside to produce it. Later, we selected the optimization parameters, again the inoculum, that in that case, instead to separate between one biomass and the other, we decided to use the ratio between them. And the second one is to tryptophan production rate, because of course we don't know which is the best rate for production based to be stable the system. Again, for biology in production and the carbon source that we wanted to use it. And the fitness, we use it, the biology concentration. What the results that we obtain? We obtain that the best solution for rate with ratio is 29 versus 1. In the paper, they identify 19 against 1 in terms of an amount of the tryptophan producer versus the biolin production. We identify as top result the galactose as the best carbon source for production, the same than the authors, and the thing that it was varying a little bit related with the flux ratio of tryptophan and biolacine, because in our case the ratios is and they are a little bit higher than they measure in the project, but of course not always are related with metabolic fluxes, maybe are related with other regulatory interactions, but the results are relatively similar to what we expected. So we are happy with the results, and in that case, we wanted to see not always what's happening with the top one. We wanted to check the rest. We again check the correlation between the production and the rest of the variables and expected with best ratio. In that case, it's a bit confusing because in that case the ratio is I inverted in the two figures. So in the ratio is put it as minus. Is because the ratio for FLYCOP I put it the biology productor and the tryptophan, and all of ratios are negative. So in the case of the carbon sources, I checked it what's going on to see if how the best results are related. If we see, for example, related with the biologine production, not only the top one of galactose are good, there are several configurations that we see in this biolin plots that are representing the top 100 values that we obtain, configurations. Okay, it means that, for example, there are a lot of results in biolin production for galactose and glucose. And the rest of them, we can see that the amount of best solutions, they are similar behavior in terms of production, but they are good enough, both of them. Related with the glucose, we didn't check it in the literature if it's true that it can be a good carbon source, but it's what our analysis can tell us, that in the future we'll check it again with only glucose. And this is the things that we can define with FLYCOP. Of course, we can increase the complexity. We can use it for much more microbial consortia. This is based on a bottom-up engineering process because, of course, you needed the metabolic models, and this is a handicap because to have nice predictions, you needed really good metabolic models, and there are not so many because not so many people used to generate good quality ones. But at least the most used species as yeast, E. coli, Pseudomonas putida, you will have it among others. So here I left the two GitHubs that you can use it for FLYCOP and FLYCOP 2. FlyCop will, in the moment that FlyCop 2 is will be published, we'll just deprecate FlyCop because it requires a very old versions of the software, but it's Dockerized, so suppose that it's not an issue about that. Well, if you have questions, I will be happy to answer.